Hello, I'm Janvi Mishra from SYBSC and I'll be presenting my assignment. The topic of my assignment is a class of biomolecules known as lipids. Biomolecules. What are biomolecules? Biomolecules or biological molecules refer to chemical compounds that are naturally synthesized by living organisms and are integral to sustaining life and executing various physiological functions. These molecules are involved in a spectrum of biological processes, including cellular replication, morphogenesis, and ontogeny. Biomolecules can be classified as macromolecules, of which are polymers. These are large, complex molecules composed of repeated subunits. Proteins are polymers of amino acids that perform catalytic, structural, regulatory, and signaling functions. Carbohydrates are polysaccharides involved in energy storage, molecular recognition, and structural roles. Lipids are hydrophobic molecules including fats, oils, and sterols that are key to membrane structure and energy metabolism. Nucleic acids contain DNA and RNA, which are polymers of nucleotides, serving as repositories and transmitters of genetic information. Second classification is low molecular weight compounds. Primary metabolites are small molecules such as amino acids, fatty acids, nucleotides, and monosaccharides, which are directly involved in essential and metabolic pathways. Secondary metabolites are specialized organic compounds such as alkaloids, flavonoids, terpenoids that are not directly involved in growth or reproduction but may serve ecological functions like defense or signaling. Natural products are structurally diverse compounds biosynthesized by living systems, often possessing pharmacological or ecological relevance. Biomolecules play a central role in maintaining and regulating the life of cells. Proteins, especially enzymes, control the speed and efficiency of chemical reactions that are vital for energy production, growth, and repair. Nucleic acids like DNA and RNA store and transmit genetic instructions, guiding how cells divide and develop. Carbohydrates act as fuel and help with recognition between cells, while lipids make up cell membranes and help carry signals inside and between cells. Altogether, biomolecules form an integrated network that keeps cells stable, responsive, and able to carry out essential life processes. A single biomolecule will be examined in depth, that is lipids. Now, what are lipids? Lipids are essential part of living cells. The term lipid comes from the Greek word lipos, meaning fat, but not all lipids are fat. The group also includes oils, waxes, phospholipids, and steroids. They are the main builders of biological membranes and create the barrier that separates the inside of cells from the outside environment. Their unique structure with hydrophilic head groups and hydrophobic hydrocarbon tails enables phospholipids and cholesterol to come together and form bilayers. These bilayers keep the membrane stable, regulate what passes through, and facilitate processes like endocytosis, exocytosis, and vesicle trafficking. Characterized by their hydrophobic or amphipathic nature, lipids are largely insoluble in water but soluble in organic solvents such as chloroform and ether. These property distinguishes them from other biological macromolecules like proteins, nucleic acids, and carbohydrates, which are typically hydrophilic. Lipids are synthesized mainly in the smooth endoplasmic reticulum of cells, while membrane proteins are synthesized in the rough endoplasmic reticulum. Some lipids, such as essential fatty acids, cannot be synthesized by the body and must be obtained from the diet. The biological function of lipids are as varied as their chemical structures. In many organisms, fats and oils serve as the main form of energy storage. Phospholipids and sterols are important structural components of biological membranes. Additionally, although some lipids are found in smaller amounts, they perform vital roles as enzyme cofactors, electron carriers, light absorbing pigments, hydrophobic anchors for protein molecules that assist in the proper folding of membrane protein, emulsifying agents in the digestive system, hormones, and intracellular signaling messengers. The structure of lipids contain core components such as fatty acids, glycerol backbone, or a steroid ring sometimes. Fatty acids are the suite of molecular chains that provide hydrophobic character to a lipid. The acidic terminal carboxyl group serves not only as the acid generating site but also as the site for esterification. The hydrocarbon chain of 12 to 24 carbon atoms contain a non-polar water-resisting tail. The chain length may vary, such as a short chain, medium chain, long chain, or a very long chain. Short chain contains less than 6 carbon atoms, which are more soluble in water and are quickly metabolized. Medium chains are absorbed and transported directly to the liver. Long chains preponderate in membrane lipids and in energy storage. Very long chains are found in specialized lipids like sphingolipids. It can also be classified as saturated, monounsaturated, polyunsaturated, or trans fats. It acts as an energy reservoir. It also improves membrane fluidity, and it also acts as a signaling precursor. Coming to the glycerol backbone, it contains three carbon atoms that constitute the backbone of glycerol, responsible for fatty acid placements and head group attachment. The three hydroxyl groups at SN1, SN2, and SN3 position can undergo esterification or etherification. Stereospecific numbering or SN ensures the lipid names remain consistent and may enable biological recognition. Triglycerides contain all three hydroxyls, which are esterified by fatty acids for energy storage. Glycerophospholipid contains two fatty acids at SN1, SN2, and phosphate-linked head group at SN3. Plasminogens confer oxidative resistance to neural tissue with an ether linkage at SN1. Functional implications. Due to the diversity of head groups, different polar groups on the phosphate generate multiple membrane properties. Uninterfered with positions of an unsaturated chain, mostly at SN2, controls the level of membrane curvature and thus protein interaction. Now coming to the polar head groups, which are attachment of polar, typically charged groups which convert non-polar diacylglycerol into amphipathic phospholipid or glycolipid. Some common phospholipid heads include phosphatidylcholine (PC), which is a zwitterion, which is prevalent in the outer leaflet of the eukaryotic membrane; PE, which is smaller in size; PS, which contains net negative charge. It does flip-flop to signal apoptosis when outside; PI, which scaffold for the phosphoinositide signaling mechanism. Glycolipid heads contain sugar moieties such as glucose, galactose linked to the glycerol or sphingosine backbone. Gangliosides consist of complex oligosaccharides with sialic acid and are essential in neuronal cell recognition. Amphipathicity and membrane behavior. Hydrophilic head groups are exposed to the aqueous environment, hydrogen bonding with water and ions. Charge wants to attract proteins that are specific. Example, pH domain actually binds to PI. Size and charge of head groups determine thickness, curvature, and domain formation of bilayers. Steroid rings. Unlike glycerolipids, sterols are not built upon a glycerol backbone, but rather on a rigid polycyclic core. Cholesterol contains an alkyl side chain at C17, which orients into the bilayer hydrophobic core. Hydroxyl group interaction at the lipid-water interface orders the fatty acid tails of neighboring lipids. Modulated membrane fluidity. Stiffening occurs at high temperature, whereas it prevents solidification at low temperature. Biological derivatives. Bile acids are oxidized steroids with several hydroxide and carboxyl group for fat emulsification. Steroid hormones have oxidation of ring A or cleavage of side chain, which leads to the synthesis of cortisol, testosterone, and estrogen. Now coming on to the classification. Classification of lipids can be done as simple lipids, which are further classified as fats and oils and waxes, compound lipids such as phospholipids, glycolipids, etc., and derived lipids such as alcohol, fatty acids, sterols, etc. Let's have a deeper look on. Fatty acids are carboxylic acids with a long aliphatic chain that may be either completely saturated or unsaturated. The terminal carboxyl group provides the acidity, while the hydrocarbon tail provides the hydrophobic character. Physically, the chain length and degree of unsaturation have an impact on properties like melting point and fluidity. In the case of membranes, saturated fatty acids pack together tightly to increase rigidity, whereas the kinks of unsaturated chains increase fluidity. Fatty acids are used biologically as energy reserves, building blocks of membranes, and they can serve as precursors for eicosanoids. Glycerolipids. Glycerolipids are the esters of glycerol and fatty acids. Phospholipids. Phospholipids consist of a glycerol backbone, two fatty acids, and a head group extended by a phosphate group. The amphipathic structure of phospholipids drives them to self-assemble as bilayers, which is the overall form of all biological membranes. The head group of phospholipids can create differential curvature, charge, and establish selectivity for interaction with proteins. Sphingolipids. Sphingolipids contain a sphingosine backbone as opposed to glycerol. Ceramide sphingosine N-acylated with a fatty acid is central hub. The various derivatives are sphingomyelins, which are phosphocholine linked to ceramide and constitutes the principal component of myelin and plasma membranes. Glycosphingolipids, which contain one or more sugar linked to ceramide, responsible for important cell-to-cell recognition processes in the nervous system and often act as receptors for microbial toxins. Sphingosine 1 phosphate, a phosphorylated sphingosine base primarily regulating cellular growth, survival, and migration. Not surprisingly, dysregulated and sphingolipid metabolism is the basis of a range of disorders from neurodegeneration to cancer. Sterols. Sterols are technically four-ring polycyclic lipids, with cholesterol being its animal archetype. The presence of a hydroxyl group at C3 gives the sterol a mild amphipathic character, as the OH group of cholesterol can face towards the membrane surface and the rigid hydrophobic rings can orient between the fatty acid tails of phospholipids. Consequently, cholesterol can alter the fluidity of membrane in two ways. At high temperature, it limits the lateral movement of the phospholipids, and at low temperature, it prevents the phospholipid from closely packing. Moreover, cholesterol is a precursor for steroid hormones such as cortisol, testosterone, and estradiol, bile acids, which are emulsifiers for dietary fats, and vitamin D. Glycolipids. Glycolipids are lipids that have carbohydrate head groups covalently attached. Glycolipids belong primarily to two classes, glycosphingolipids and glyceroglycolipids. Waxes. Waxes are esters of long-chain fatty acids and long-chain alcohol, forming solid matter that is highly hydrophobic at room temperature. In nature, waxes cover plant leaves and fruit to prevent evaporation and protect against pathogens. Waxes insulate and waterproof animal fur, feathers, and skin. Waxes are energy storage molecules in zooplankton and marine animals. Their resistance to oxidation and water, together with being solid at room temperature, makes them ideal biological barrier materials. Lipoproteins. Lipoproteins transport lipids in an aqueous environment of blood and lymphatic fluid. They have a core made of hydrophobic lipids, cholesterol esters, and triacylglycerols, surrounded by a shell made of phospholipids, free cholesterols, and apolipoproteins. The main classes of lipoprotein organized by density from low density to protein lipid ratio to high are chylomicrons, VLDLs and LDLs, and HDLs. Chylomicrons transport dietary fats from intestine to tissue. They are high in triacylglycerols. VLDL transport endogenously synthesized tags from liver to adipose and muscle. LDL delivers cholesterol to peripheral tissues. Elevated levels are associated with atherosclerosis. HDL, reverse transport of cholesterol from tissue back to liver for excretion. Good cholesterol carrier, they are considered as a good cholesterol carrier. The transfer of apolipoproteins and lipids on LCAT and CETP modulates their metabolic rate and activity. Sacharolipids. Sacharolipids are lipids where fatty acids are directly attached to a sugar backbone rather than glycerol. The prototypical example is lipid A, the glucosamine-based anchor of lipopolysaccharide in gram-negative bacteria, comprised of a disaccharide of glucosamine N-acylated with multiple fatty acids. Sacharolipids associate with bacterial outer membrane and can induce host immune responses as endotoxins. Now let's look at the biological roles of lipid. Structural role in cell membrane. Phospholipids constitute the bilayer structure of biological membranes, offering selective permeability and providing fluidity. Cholesterol intercalates between phospholipids to adjust the rigidity and fluidity of membrane, especially for animal cells. Sphingolipids partake in the formation of lipid rafts, which are highly specialized microdomains of the membrane that participate in signal transduction and protein sorting processes. Second, energy storage and metabolism. Triglycerides stored in the fat tissue are the primary long-term energy reserves, providing around 9 kilocalories per gram. In conditions of fasting or exercise, lipolysis liberates fatty acids which enter the beta oxidation pathway in mitochondria and finish as acetyl-CoA, which enters the citric acid cycle to make ATP. Ketogenesis forms ketone bodies as alternative fuel in conditions where glucose is scarce, especially for the brain. Third, hormone and signaling molecule precursor. Steroid hormones such as cortisol, estrogen, and testosterone are generated from cholesterol and are involved in the regulation of metabolism, reproduction, and stress responses. Eugosinoids such as prostaglandins and thromboxanes derived from arachidonic acids mediate inflammation, vasodilation, and platelet aggregation. Lipid signals include molecules like diacylglycerol and inositol triphosphate (IP3) that activate intracellular signaling cascades. Nervous system functions. Myelination, the wrapping of myelin sheaths around axon, contain around 70% lipids and allow for the rapid conduction of nerve impulses. Lipids also have some influence on neurotransmitter release, synaptic plasticity, and cognitive function. Omega-3 fatty acids and DHA are important for brain development and mental health. Insulation and organ protection. Subcutaneous fat provides thermal insulation to the body in maintenance of thermal homeostasis. Visceral fat provides cushioning around the internal organs and protect them from mechanical shock and bodily injury. Fat pads in joint surfaces and the soles of feet minimize friction and impact stress. Transport and absorption of nutrients. Lipids enable the absorption of fat-soluble vitamins such as A, D, E, and K. They also enhance the bioavailability of several phytochemicals like lycopene and beta-carotene. Metabolic regulation and homeostasis. Adipose tissue secretes leptin that regulates appetite and energy balance. Lipids also affect insulin sensitivity, glucose metabolism, and inflammatory responses. Transcription factors like SREBPR and PPAR regulate lipid biosynthesis and oxidation. Lipoprotein transport in blood. Because lipids are hydrophobic, they are transported via lipoproteins. HDL or high density lipoprotein removes excess cholesterol from tissue. LDL, low density lipoproteins, delivers cholesterol to cells but can contribute to atherosclerosis if elevated. Studying lipids involve a combination of extraction, separation, and analytical techniques tailored to their chemical diversity and biological roles. Here's a detailed breakdown of the most widely used methods. Lipid extraction techniques. Extraction of lipids is the first and critical step in lipid analysis and is the process of isolating lipids from a biological matrix with the least degradation and contamination. Classic solvent-based methods. Falt method. This method uses chloroform methanol to produce total lipids from tissues. It can be used effectively on a solid sample. Bly and Dyer methods. This method uses chloroform methanol water to obtain wet samples. When the complete extraction occurs, it produces a monophasic system. And once the extraction solution is added, it will have a biphasic appearance for the separation. Solute extraction. This is continuous solvent extraction in a dry state. Solvent choice are usually hexane or petroleum ether. Contemporary and environmentally conscious extraction methods, supercritical fluid extraction or SCFE. This extraction method, which uses supercritical carbon dioxide and frequently employs ethanol as co-solvent, is very effective for removal of omega-3 fatty acids and sterols with reduced oxidation. Enzyme-assisted enzyme-assisted extraction. It uses various lipases, proteases, or carbohydrates to disrupt cell walls and liberate lipids, particularly from microbial or plant sources. Microwave-assisted extraction and ultrasonic extraction. These processes aid in solvent penetration through material and reduce extraction time. The appropriate lipid analysis relies on effective separation techniques that distinguish lipids based on polarity, volatility, and molecular structure. In lipid analysis, classic solvent-based procedures like solid-liquid partitioning and solid-phase attraction are often applied to prefractionate lipid extracts into neutral lipids, glycolipids, and phospholipids before chromatographic separation occurs. Once lipids have been prefractionated and or desalted using these techniques, chromatographic methods can then achieve high-resolution separation and analysis. Normal phase and reverse phase liquid chromatography, gas chromatography, and thin air chromatography are still the most widely used separation methods, while new separation methods such as supercritical fluid chromatography and counter current chromatography also offer greater specificity for larger and complex lipids. Thin layer chromatography for separating lipids on silica or alumina. Thin layer chromatographic plates. The method relies on lipid traveling through capillary action within a solvent system that was designed specifically for the lipid polarity. Once lipid development is complete, the lipids are visualized by exposing the TLC plate to iodine vapor or fluorescence and quantitate using densitometry. Although not as sensitive as hyphenated method approaches, TLC is inexpensive and quick turnaround, making it a useful tool for screening lipid class composition and to track purification schemes. Gas chromatography is a powerful technique for profiling the volatilized lipid derivatives, particularly for fatty acid methyl esters and sterols, with lipid extracts being derived generally methylated with BF3, methanol, or acetylated before being injected onto capillary columns. Volatilized lipid separation occurs based on boiling points and polarity, while FID or MS determines the lipid's identity. GCMS or gas chromatography mass spectrometry continues to be an excellent means of resolution for determining different chain length and unsaturation isomers, enabling the quantitative fatty acid profiling for lipid extracts or biological samples. Liquid chromatography, high performance LC, HPLC, and ultra performance LC, which is UPLC, offer additional means for hydrophobicity and head group polarity analysis of intact lipids. While normal phase liquid chromatography produces good resolution of phospholipid classes, many researchers use reverse phase LC to discriminate between molecular species based on acyl chain length and saturation. Additionally, LC can be coupled to charged aerosol detection or evaporative light scattering detection (ESSD) for class-wide quantitation, or tandem mass spectrometry for a lipidome-wide targeted quantitation of various lipid mediators. Countercurrent and supercritical fluid chromatography (CCC, CPC, or SFC methods can separate lipids via biphasic and supercritical fluid characteristics, respectively, which do not require solid support. CCC or CPC works as a column and uses two liquid phases. The second is manipulated into circulation and a stationary liquid phase. Induces separation based on partition coefficients between two liquid phases. And if SFC method uses contribution from supercritical CO2, often in combination with ethanol, to causally affect the polarity of the liquid and enable selective extraction of triglycerides, sterols, and phospholipids under gentle extraction conditions. Often, initial methodologies appear to invoke temperature degradation and residual solvent issues, making CCC, CPC, and SFC more suited for selected subclasses of lipid species, such as omega-3 fatty acids and oxidized lipids that require extra care during extractions. Mass spectrometry-based detection. Mass spectrometry has become the preeminent technology for lipid analysis. Mass spectrometry offers flexibility, sensitivity, and specificity. A shotgun approach to mass spectrometry can profile lipids by means of precursor ion and neutral loss scans to comparatively quickly survey hundreds of lipid species with direct infusion. The incorporation of liquid chromatographic separation prior to electrospray ionization is a desired improvement of mass spectrometry analysis of lipids. Structures can be elucidated via collision-induced dissociation, fragmentation of iota groups in combination with accurate mass and high-resolution measurement of the lipid ion. Additionally, the use of tandem MS and even ion mobility spectrometry may aid in resolving isobaric lipids. Finally, absolute quantitation is possible with stable isotope labeled standards in complex matrices. Spectroscopy and complementary techniques. Nuclear magnetic resonance (NMR) spectrometry provides functional group identification and even quantitation of lipid species without derivatization and with a level of absolute structural information. However, when using NMR, users need to be aware of the need for milligrams of sample. Infrared and ultraviolet visible spectrometry, also known as UV-vis spectrometry, provides additional methods for functional group screening and conjugated double bond quantitative assays. These spectroscopic methods combined with chromatographic methods and increasingly mass spectrometry add to an extensive analytical toolkit for lipid-based research, quality control analysis, and biomarker discovery. Thank you.